Good afternoon, everyone. Thank you all for joining Alchemy's monthly webinar series. So just for those of you who don't know, Alchemy is a UK hub funded by EPSRC with the goal to kind of pioneer and normalize the use of AI for chemical research. So today we've got two speakers lined up, each sharing very insightful talks, and then we'll have a Q&A session after each presentation. So please feel free to use the Q&A function to ask your questions during the talk or at the end. So I am the host for today. I'm a lecturer in the Department of Chemistry at Imperial College London, and I'm very excited to introduce our first speaker today. So this is Linden Schrecker, who is the CEO and founder of Solve Chemistry. So Solve Chemistry is an Imperial spin-out focusing on enabling efficient chemical process development through automated high-throughput data collection and machine learning. So Linden did his undergraduate degree at the University of Oxford, after which he moved to Imperial to complete his PhD, which was kind of split over chemistry, chemical engineering, and machine learning. So his PhD research focused on developing a new methodology to efficiently collect data from chemical reactions. So I guess we're going to be hearing more about that today. And his PhD produced many peer-reviewed papers, along with a patent, and it was awarded departmental thesis and presentation prizes. So since founding Solve, Linden and his team have further improved the data collection techniques and are now working with many industrial clients. So I can hand over to Linden now.
Great. Thanks very much for the introduction, Alex, and thanks, Alchemy, for inviting me along to present here also. So, as Alex said, what we kind of focus on is efficient data collection for efficient decision making, specifically around process development. So a little bit different to some of your other kind of AI or data collection chemistry startups. We care about the process development stage, not about the actual molecule, but about the process conditions. So as was mentioned, we're an Imperial spin-out funded by a UK-based venture capital company, Creator Fund, who many of you may have heard of, and Chemovator, who are a BASF-aligned venture fund as well. And we've done work, as you might expect, for BASF, for Alchemy, and then for others as well, and people we can't kind of mention here as well. We've had a range of nice very thankfully glowing articles written about us by various people, which is very nice as well if you'd like to find out more about our kind of backstory in different ways also. Okay, so as I kind of alluded to in the introduction there, what we care about isn't so much the actual drug or chemical that we're producing. There are lots of others that do this very well. The big ones at the moment kind of being isomorphic labs, for example, where you're predicting what drug structure you might want for treating a certain disease, or from an agrochemical perspective to solve a certain agricultural problem. There is also significant amounts of data, both in the literature through the likes of the Open Reaction database, kind of fronted by Connor Cooley, and then also within many different companies, for example, AstraZeneca, Pfizer, Syngenta, BASF, all having large data sets of the A+Bs that you use to get to certain products as well. So these are the kind of retrosynthetic planning tools. We have a nice load of data around this and decent machine learning methods for doing this, some of which are open source, such as AI Zinth, for example, as a kind of front runner there coming out of the kind of ASCOS engines. What we care about is the red question mark over the arrow and how this affects scale up. So once you've made a milligram of your compound, whatever chemical you want to make, and you've done it kind of quick and dirty as you have every right to do in these early stages, you then need to start thinking about how we're actually going to make a kilogram, a ton of this stuff in a meaningful, efficient, sustainable, economic manner. And that's where these start to come in a lot more, and how we design this process. So a lot of continuous high dimensional parameter spaces here. So as I mentioned, despite there being large data sets around kind of products and around A+Bs, there's surprisingly little data around how different continuous variables and process conditions affect the outcomes of these reactions in order to build kinetic models, process models in different ways. This broadly seems to come from within industry, a want to do optimization of each process as quickly as possible, and therefore trying to do it in as few data points as possible. This is great if you're just trying to optimize one process, but if you're trying to optimize loads and loads of processes and move the kind of the number of data points you need per process long term to a shorter, lower number, then it is more useful to have a lot more data. So I think many of you may have experienced the situation before where you end up having great data for what they were trying to do at the time, but the data is not very useful for later on. It's also very inefficient to collect data for these process conditions and quite hard to computationally model this without the data. This actually gives us a very nice opportunity for what our investors have started terming full stack chemistry, which is a bit buzzwordy, but maybe gets across the idea quite nicely, that we have an opportunity where there isn't much data to integrate automated data collection for this area, so build the data set whilst doing the machine learning at the same time. So we're not relying, as other areas of chemistry often are, on data that already exists and then doing machine learning on suboptimal data sets. We can create the right data set whilst doing machine learning at the same time, which is nice to be able to do. The benefits of this are generally that we get more drugs and agrochemicals faster and cheaper, but we have benefits to do with de-risking kind of capital expenditure, so which equipment you build for doing this scale up, what kind of conditions you run this under, and reducing the costs and gaining sustainability from this, shorten time to market by collecting this data faster that you would otherwise collect, and more of it in a more robust manner, so we can make decisions at the same level of risk but faster, especially in the pharmaceutical space, and broadening out now into supporting these processes for regulators as well. So using this to, if you can't find a more sustainable way, for example, being able to justify that you've tried very hard to find this, and noting where the limitations and the trade-offs are for this. Our approach stems from some of my PhD work in the middle here, which is how we collect this data in the most efficient manner using a technique called transient flow, which I'll introduce in a minute. And then with my co-founder, Jose, who did his PhD under Ruth Meisner in computer science in Imperial, on not just how we collect data in a really efficient manner, but we need to be collecting the right data. So we need to plan this experimental design through. You can think of this as DOE on steroids, but broadly it's incorporating a little bit more kind of chemical expert knowledge there, and it's thinking a bit more broadly about how we minimize uncertainty across the entire space instead of just trying to do optimization problems. So we're designing which experiments we do in the most effective manner. We're collecting data in the most efficient way, but really our value is not in the actual data points itself, but the insights these produce. So we do a lot of transfer learning and other ML on the back end as well to try and get the most information possible and the most insights out of each data point we produce also. Okay, now for an intro on transient flow as an actual data collection technique with a few examples floating around as well. So traditionally in chemistry, we collect data in batch. So we set up a round bottom flask or a vial. We leave it under a certain set of conditions for a certain amount of time at a certain temperature under certain concentrations with a catalyst and a solvent. And then we work it up, we analyze our yield, and we get 1 point out of each data, out of each experiment, which seems very wasteful to me in general. Luckily, for kind of quite a while now, for a few decades, we've been doing a lot of in situ analysis and sampling. This naturally gives you time series data out of a batch reaction. You set it up under certain process conditions, and then you can monitor it over time and see how it progresses, which is great. So you've massively gained the efficiency of how you collect this data. In flow, we can get more accurate data than we get in batch, as we have better mass and heat transfer generally. So we're getting more accurate knowledge on the precise conditions that were used in a reaction, and we can have a broader range of conditions as well. So we can go to supercritical conditions easily. We can go to very short reaction times very easily as well. However, we're back to just doing endpoint analysis and gaining 1 data point per experiment, as we need to leave the system to settle under certain concentrations, flow rates, temperatures, and then collect a data point before we change this. However, if we vary flow rate, the cumulative flow rate of the system in a well-controlled manner, we can get a time series of data out of this, essentially as we're looking at the volume of the reactor divided by the flow rate in order to get to the reaction time. If we have a variable function of flow rate, and have a fixed volume, we can get time series data out of this, and we can deconvolute this in real time. So that gets us to the efficiency of a batch reactor with an in situ analysis probe again, but maintaining the advantages of flow chemistry and some of the disadvantages as well. For example, solids handling, which is challenging in flow generally when we collect this data. However, the real advantages come when we start applying this technique to other variable parameters as well. And we can do this with variable temperature, reactants, stoichiometry, additive concentrations, solvents, and then applications in kind of photochemistry and electrochemistry that you might be able to imagine as well on continuous parameters there, which allows us to scan this design space much more efficiently and get continuous data, even at fixed time points or by varying multiple ones at the same time. As a quick example of that, here's the Knorr-Pyrazole synthesis. So it's a double condensation of this 1,3-diketone with a hydrazine here. We go reversibly by this hydroxypyrazole intermediate to form, then dehydrate to form the pyrazole product. This was the broadly reported mechanism that everyone's been very happy with. We collected a nice load of data over this space with reactants stoichiometry ramps, so varying the ratio of 1 and 2 and varying the residence time. And we found that the kinetic model of this was nowhere near fitting the actual data we could produce from this. It was a massive oversimplification. And because of the amount of data we collected, we had 600 data points collected in about a day here, using inline React IR in this case, we could robustly fit a much more complex microkinetic model with a high degree and quantified uncertainty in these different metrics as well, which was very nice to see. Another application case is when we look at selectivity of processes. So in this case, we're looking at part of a chemoenzymatic dynamic kinetic resolution process. So we're trying to racemize this amine. So we're trying to make the E go down whilst keeping the selectivity high. However, it's not as simple as a multi-objective optimization problem in which you could just kind of look at the Pareto front between these two trade-offs, as we have another approach where we can recycle this around the system as many times as we like. So broadly, we're looking at a slightly more complex optimization problem. So for scale up, we were looking at actually just collecting a kind of black box model out of how temperature, flow rate, and solution concentration affect the E and selectivity of this process. And you can see how this is affected by a temperature ramp here. E getting lower with temperature as we want to see, but selectivity also dropping with temperature after an initial plateau at these conditions. We don't just do one factor at a time in this, though. We scan the design space nice and robustly to look at how EE is affected by these different parameters across the design space and how selectivity is as well, giving us more insights into how we can scale this up. And this process was then scaled up into a kilo setting within BSF also. Solvents is where it starts to get particularly interesting. Solvents being an important thing for many reactions, but not being in the end product. So a lot you can kind of play with here, but often very harmful and making up. I can't remember the exact stats. I'm sure Nesta can correct me on these later on. But I think AstraZeneca says that it makes up something like 80% of the CO2 emissions of the actual process is made up of the solvent, with only 20% being made up by other parts. For this, we chose this simple paracyclic rearrangement, so an aromatic Claisen rearrangement, which was known to have considerable solvent effects. So no one really wanted to say precisely what these solvent effects were. And quite a few computational groups had a stab at this, but with minimal data. We collected a lot of data over different binary solvent mixtures in order to explore this design space. So for example, here going from 100% ethyl acetate, going via lots of incremental 1% kind of changes, say, into pure methanol, going from methanol into 50% water in methanol, and so on and so forth, with a lot of different solvent ramps. We can then parameterize up these different solvent mixtures using a range of different ways of parameterizing this in different ways. Here was kind of 13 standard, some computationally derived, some empirically derived parameters, and we can see that none of them really fit perfectly. Those things like ET30, which is a empirical measure of polarity, work quite well. We can then train a machine learning model on these 250 solvent mixtures and find many different suitable replacements to the normally used DMF for this system. However, what you might be thinking is, how did you actually choose those solvent ramps? Solvent space is very, very large. So this comes into one of the other pillars, more on kind of Jose's side of this, about how we actually do the experimental planning to make sure we're doing the right experiments. This goes into some work we did for alchemy, nice and fittingly, where we were looking at a slightly more complex aromatic paracyclic rearrangement reaction, where you have this starting material that goes via some hidden products to this product, and then to this product as well. And we can see the trade-off in a residence time, temperature, solvent space here, where we're collecting this for each kind of solvent ramping example also. What we first need to do is define a solvent search space, so know where we're actually looking. We chose the kind of standard space from the AZ solvent selection guide that people standardly use for this. And then what we do is we predict the uncertainty in all the kind of ramps, all the different conditions across that space. We initialize with a ramp that will go across a lot of this space to give us the most information, and then we retrain our model and predict the uncertainties on all the other kind of ramps we might have done in that space. Then we choose the one which has the highest uncertainty, so that when we perform it, we gain the most certainty on this in a kind of Bayesian manner. So in this case, we'd then run this HFIP to IPA ramp. We then retrain and run again until we have minimized uncertainty across this entire design space. How this looks, in this case, represented in this kind of PCA space here, but the space obviously that we're exploring is the high dimensional one in this case. We're exploring through the space between different solvent mixtures, and then we can use this to predict various green solvents, such as styrene and ethyl lactone and things like this, using these featurized solvent mixtures that we've previously performed. On how we do our standard data collection, we're not doing this manually, we're doing this in a very automated system, and we're trying to use it in an automated way to gain the insights as quickly as we can. How this generally looks is a reaction comes to us, we define a high dimensional parameter space broadly around these kind of parameters. Our algorithm designs an optimal experimental campaign through this space, looking at ramps we perform through this space. So instead of individual data points, we're getting lines of data through the space, and we can vary where these are going. For example, in the temperature and different flow rate regimes here. We can turn this into automated commands for our kind of automated flow system, and we can monitor how these vary through a web-based portal run through weights and biases stolen from the machine learning community, but very good for looking at instrument actuals, and monitor things like the pressure and the HPLC results as they come out of this as well. So we can online analyze all the species that are in our reaction and deconvolute what conditions these were collected under. We can then plot these on, in this case, a three-dimensional space to see what the trade-offs are between different parts of this space. And we can train models on this in various ways, ranging from fully black box models to fully microkinetic models. But broadly, we try to sit somewhere in the middle, as what we really care about is doing the best predictions across a defined design space, not on being stuck to any particular method. And this allows us to look at the different trade-offs between process conditions and reaction outcomes. For example, for this cross-coupling reaction here, we are encoding a bit of expert knowledge in advance, in this case quite basic expert knowledge, which is when you increase the temperature, the reaction is probably going to respond in a Arrhenius type behavior, so have an exponential effect. And this means that even though we didn't collect data in this higher bit of the space, we are still being chemically consistent, and we can extrapolate this data well with well quantified uncertainty. When it comes more broadly to which A plus Bs we are actually performing in this space, we try and collect data that best informs the rest of the space around it. So it's not being limited to the individual reaction that it's being collected on, but can inform other reactions around it. Particularly important when we're looking at more expensive, hazardous and IP sensitive reactions. We can collect reactions around that in the chemical space and use that to inform the target reaction of interest instead. For example, in this cross-coupling, we're taking different A reactants and B reactants together with different R groups and functionality associated with them. And if we trained on 8 of these, we could well predict, this is a leave one out study, so we could generally well predict the outcomes across that entire kind of temperature, time, concentration design space for the other examples as well, which is very exciting. This allows us then to work out to cover a certain entire chemical reaction class, how many reactions we need to do and how much data, and where we should be placing these to have a high degree of certainty across the entire design space. That's just left to say, currently we do a lot of data collection projects based around mechanistic and kinetic modeling and solvent understanding, though we do a range of other ones here as well, with our first service contract signed with BASF this month, working on querying of our automated system as often as they would like, given a certain capacity. And if there is other data around, we can work out what data we need to improve the predictive performance of these data sets also, and fill that in. We work in a variety of ways, depending on what your kind of role is in the audience, with industrial contracts, academic collaborations, to aid translation of chemistry in academic settings into industry by helping collect that data, as well as doing co-development partnerships with a variety of reactor type companies in order to assess new reactor types and prototype new systems. It's not just me that did all this. I'd like to thank Jose, Sarah, and Tom as well in the team working hard on the machine learning side and on the automation science side over in Scale Space, if anyone would ever like to visit, as well as postdocs from the Iconic side and various others on the machine learning front and commercial side, helping us make Solve what it is today. And with that, thank you very much.